Welcome to AP Biology. Today I want to talk to you about the structure of DNA and RNA. So the monomer for DNA and RNA is called a nucleotide, and the nucleotide is here, or if you look at it here, it's right there. It's a fairly complex nucleotide, a fairly complex monomer because it's made out of three parts, a sugar and a phosphate and a base. So the nucleotide is made out of a sugar, that's this. Sometimes you'll call it a pentose sugar because it's got 5 carbons or 5-carbon sugar. There's a phosphate group, that's this, and then there's a nitrogenous base or just a base. There are, the sugar is deoxyribose in DNA and ribose in RNA, so deoxyribose is just missing in oxygen. The phosphate group is the phosphate, oops, wow, I've struggled here a little bit. The phosphate group is here. Let's go back to this. Here's the phosphate group. Okay, the base. So the nitrogenous base varies. In DNA, it's adenine, thymine, cytosine, and guanine. In RNA, you use uracil instead of thymine. So for DNA, the bases are A, T, C, G. For RNA, the bases are almost the same. It's A, U, C, G. So there are two kind of big classes of groups. There are the purines and there are the pyrimidines. The purines have the shorter name, but the molecule's bigger, so it's a 2-ring base, whereas the pyrimidine is the longer word and it's got only 1 ring. What you really need to know is that the purines bind with the pyrimidines to keep the radius of the diameter of the double helix the same. So here's a DNA molecule, but if you were to untwist it, it might look like this. And if you had 2 big bases binding, the diameter would be so big here that it would be stressing the bonds. Or if you had 2 little bases binding, maybe the diameter would be too small. So instead you have a big one and a little one bind, or this big one and this little one. So the DNA polymer is a double strand of nucleotides in a twisted ladder shape. It is not always double-stranded. There are viruses that have single-stranded DNA, but most of the time that we're talking about it, you can remember DNA is double-stranded. So the sides of the ladder are alternating sugar and phosphate groups, and they are held together by the strong covalent bonds. If you look at this picture, I would want you to be able to circle a nucleotide for me. I would want you to be able to tell me this is the sugar. Here's the phosphate group. And here is the base. So this thing that I circled is the nucleotide. I would want you to be able to show me that the bases bond with each other here in the middle, and that you have a big one, the purines, with a little one, the pyrimidines. Notice always little one with big one with little one. That keeps the radius or the diameter right here. Whoops, whoa. Keeps the diameter the same all the way. So the steps or the rungs are the bases, the adenines that bind with the thymines, or the cytosines that bind with the guanines. They meet in the middle. They're held together right here. These are weak hydrogen bonds. Here, here, here. Because you need to be able to unzip it easily. And so hydrogen bonds are weak. They can unzip pretty easily. That way you can use the DNA if you need to make RNA, or you can copy it when it's time to do replication. So you can unzip it. Okay, so more about nitrogenous bases. So the purines, again, are the big ones. Pyrimidines are the little ones. Double rings or single rings. Base pairings, you always have a purine bind with a pyrimidine. So you have the big ones with the little ones, so the radius stays the same. You probably don't need to memorize this, but you do need to know the guanine and cytosine pair. The reason is that you have a big one with a little one, and also they have 3 hydrogen bonds, as opposed to adenine and thymine. Again, little one with big one, but they have 2 hydrogen bonds instead. So this is called complementary base pairing. A always binds with T, C always binds with G. You probably memorized that at one point. So if I give you a long list, I would want you to be able to give me the other side of the DNA molecule, right? And then the other thing I would want you to be able to do is if I told you that 15% of a sample of double-stranded DNA was T, I would want you to be able to tell me that 15% must therefore be adenine, and then also be able to figure out the amounts of cytosine and guanine for me. So This, that the big one always pairs with the little one, keeps the radius or the diameter of the double helix the same. Okay, next thing that I want to show you, do you see how the O's of the sugars are pointing up here, but they're pointing down here? They go in opposite directions. And so that's important because enzymes are physical molecules. There are these proteins that physically just fit one way or another. So if you picture a key going into a lock, you can put a key here into a lock, but you can't put it in the other way. It doesn't fit over here. So just like a key has an actual shape and works a certain way, same thing with enzymes. They can fit onto certain things. So an enzyme might recognize this shape right here, but wouldn't be able to recognize this shape over here. So that this is different than this matters. And that it runs in a different direction both ways also matters. Sorry, this was a few minutes ago. So if there's 30% adenine, how much cytosine is there? Well, 30% in a double-stranded DNA sample. If there's 30% A, then that means there's 30% T, which means 60% A's and T's, which means that there's got to be 40% C's and G's, which means 20% G and 20% C. Did I do that right? Yeah, 60, yeah, 20%. Yay. Okay, so now what I want to do is talk to you about the direction of the nucleotides. So here's a nucleotide. It's got a sugar and a phosphate and a base. And so I want you to look at the sugar. We're going to name these sugars, and we could have named like the sides of the sugars. Because this shape is different than this shape, enzymes can recognize one side but not the other side. And they could have called them like, I don't know, like the vanilla side and the chocolate side, but they didn't. They called them chromosome, they called this carbon 1 and this carbon 2, and they called this one 3, and they called this one 4, and they called this one 5. And you don't really need to know that, except you do need to know they all sort of line up on top of each other. So if this is the 3' side, and you put a whole bunch of nucleotides on this side, the other side is going to be the 5' side. So we're going to label these carbons, 1, 2, 3, 4, 5. And what do you really need to know? If I tell you this is the 3' side and you stack a whole bunch, I don't care if they go on for 1,000 nucleotides, on the far end, the other side will be 5. And if this side is in one direction with the O's pointing up and the O's are pointing down here, then 1, 2, 3, 4, 5. This must be the 5' and this must be the 3'. So do you see how it's opposite? If this is 3, this is 5. If this is 3, this side's 5. We call that anti-parallel. They are going in the opposite direction from each other. So this displays anti-parallelism. It means, yeah, we're parallel like in math class, but there's actually a certain direction. And that matters when you copy your DNA into more DNA or when you make DNA become RNA. The enzymes can only read one side. They're going to be able to recognize one side of this nucleotide. Okay, the bases along any strand of DNA are arranged in different orders. So it's different in a bat than it is in a planaria than it is in jellyfish. But what's the same with all of them is if you have an A, the other side has a T. So whether you're a jellyfish or a planaria or a bat, if this is an A, the other side is a T. So for every species, A always binds with T and C always binds with G. The order of bases on one side determines the order of bases on the other. So this stuff in the middle, this is actually the instructions. The bases are the actual instructions on how to build your proteins, which make you.